Okay, so today we'll be covering Vibrionaceae. Okay, so Vibrionaceae oxidase positive. So oxidase important test. It's fermentative. It's fermentative, so because it's fermentative, you are able to differentiate it from Pseudomonadaceae. Okay, because Pseudomonas in Pseudomonadaceae is always oxidative. So that is another important test. So the Vibrio in the genus Vibrio, there are more than 400 species now. Almost all of them are marine bacteria. So okay, almost all of them grow in the sea. Okay, but you have Vibrio cholera that grows in fresh water. Okay, so we have Vibrio cholera important and Vibrio parahaemolyticus and Vibrio vulnificus. Vibrio vulnificus is considered an emerging infectious emerging disease. Oh, right there. Sorry. Okay. So we are still in the class Gamma proteobacteria. As I've mentioned, Gamma proteobacteria are mostly bacteria that we can culture. Okay. So the two main tests, oxidase and OF sugar, will allow us to differentiate Vibrionaceae, Pseudomonadaceae, and Enterobacteriaceae. Okay, so this is an important test to differentiate these three main families. So Vibrios are gram-negative, motile, and curved bacterium. So a bit like a comma sign. Okay, so most of them are in the surface waters, marine and freshwater. So because of that, they are also associated with aquatic. So Vibrio cholera, okay, I'm sure you have heard of this bacteria. Is a causative agent for cholera. Cholera is actually endemic in certain states in Peninsular Malaysia. The most popular one would be Kelantan. So in Kelantan, there are a lot of wells. They use water from the well. So it seems that there is a Vibrio cholera is endemic in those waters. Okay, maybe because yeah, but Vibrio cholera can grow in fresh water. So that is probably a problem that requires more in-depth investigation. So it's slightly curved here, with a single polar flagella, found in brackish and freshwater. Brackish means the water is in between fresh and seawater. Okay, so usually you find this brackish water more in the river mouth. The river mouth, the estuary, where you have a mixture of fresh water and seawater. So that's where you get the brackish water. Okay. So in the past, Vibrio cholera is the enumeration of Vibrio cholera is straightforward. However, now we understand more about the bacteria. We know that it shrinks to a unculturable state, but still contagious. So it has a state which is called the viable but not non-culturable. So VBNC. Okay, viable but not culturable. So although that's why if you do a normal isolation procedure for Vibrio, you do not see any counts, but that doesn't mean there is no Vibrio cholera. They could have gone to a VBNC state. Okay, so you have to You have to be wary of that. So the antigenic variation in Vibrio Cholera, you have about 139 serotypes. So the most important is the SIR2001. This is the pandemic strain. This one can cause the cholera pandemic. Okay, so you have cholera, you have altor. These are the two bio and also you have the Bengal's 0139 Sirova. So the O1 is the classical strain. So whenever you isolate Vibrio cholera, uh, you should carry out a serological test to see whether they are O1. Okay, if it's O1 or O139. So if it's O1, then later on you will further serotype it, whether it is a Ogawa, Inaba, or Hikojima, according to the antigens that they have. Okay, so non-O1 strains causing typical cholera is rare, but O139 is the other one that causes cholera. So now there are two strains, O1 and O139. But O139 has only been found once. Okay, so usually it is O1 that is important. So humans are the only source of infection. Transmission is via foods and drinking water. So when Vibrio cholera is ingested, very high infectious dose, more than 10 power 8. Okay, it grows in the small intestine. It is non-invasive. Okay, incubation period for 5 days. It produces an exotoxin, Cholera Toxin, that cause the host cells to secrete chlorides, bicarbonates, and water. So what happens is, if someone has Vibrio cholera, ingested Vibrio cholera through dirty water, the cholera toxin will induce massive watery diarrhea. Okay, up to 20 liters per day. Okay, leading to exsicosis. Exsicosis means because too much water loss from your body, your blood becomes too viscous to function properly. So you'll have organ failure, your kidney and your heart. Okay, so the important uh, immediate treatment here is uh, replenishment of liquids. Okay, so the cholera toxin is it has 5 binding B subunits and 1 A subunit. A subunit has a A1 and an A2. So what happens is, usually we have a cyclic AMP that stimulates the mucosal cell to pump large amounts of chlorine into the intestine. However, this is regulated. This step is regulated. What happens when you have cholera toxin is, the cholera toxin disrupts the breakdown of the regulatory protein. Then it ends up that the cyclic AMP is indefinitely activated. The adenylate cyclase is always activated. So you the Commonly, it is the activation of this protein, the GS protein, that will activate the adenylate cyclase. So adenylate cyclase activated will produce cyclic AMP. After that, the GTPase will break down this unit to inactivate this unit, to inactivate the G5 protein. But the cholera toxin disrupts the process here. So what happens is you remain stuck at this state. Cyclic AMP, the adenylate cyclase remains active indefinitely. That means a lot of cyclic AMP is being produced. So a lot of chlorides, a lot of bicarbonates will be secreted into the intestine. And because of osmotic pressure, the water will also be secreted out into the intestine. So you lose a lot of water because of this. So lethality is up to 50% in untreated cases. So the treatment is very straightforward. Replacement of electrolytes and water, and then antibiotics to reduce the bacterial load, okay, to reduce the number of bacteria, Vibrio cholera in your intestines. So the treatment is straightforward. So why do we still have cholera epidemics? Then you have to uh, think of situations where Vibrio cholera epidemic occurs, cholera outbreak occurs. So cholera outbreak occurs after a typhoon, after mass flooding, after earthquakes. So when these catastrophes occur, the health services break down, transportation is disrupted. So water supply is also, clean water supply is not available. So when you have all these factors in play, the people in the area do not have supply of clean water. So if they drink contaminated water, they'll get cholera. And when they get cholera, they don't have access to health services. And that's why it gets worse and worse. So you have uh, mortality, people dying from cholera uh, in these areas. So usually after a calamity, typhoon, earthquakes, mass flooding. Okay, if the health services are working properly, um, cholera disease is treatable, okay, and can be controlled. There won't be any outbreak. So it's the same. Once with COVID-19, uh, the main concern is to ensure that the health services don't break down. So for the health services not to break down, you have to look at the number of active cases that we have. That's why the Ministry of Health is always emphasizing how many people is being discharged and how many new cases. Because the number of active cases have to be kept at a level that we can manage. Okay, then if you look at other countries, when the number of active cases grow too much, the health services break down, and then you have patients that don't even have hospital beds. You have patients that are not even treated. Okay, so the important thing is to ensure that the health services are able to manage the number of patients that are positive for COVID-19. Okay, yeah. So that is the main concern when we have this kind of pandemics or outbreaks. So Vibrio cholera, the symptoms are rice water stools. Okay, so rice water stools because the stool is very watery, but also you have intestinal mucus, epithelial cells, and bacteria in the stool. So it looks like rice water. So a lot of water with a bit of specks here and there. So untreated cases have 50% mortality rate, and treatment is usually replacement of lost fluids and antibiotic treatment. Next is Vibrio parahaemolyticus. So why we cover this is because it is an important bacteria associated with seafood. Okay, so Malaysia is one of the major exporter of seafood. So we export shrimps, fish, we have quite a thriving aquaculture industry. So we export to China, to America, to EU. But this America and EU are very strict. So they require testing. And one of the tests that they require is actually Vibrio parahaemolyticus counts. So you have to see whether it's present or absent in your sample. Okay. Although nearly 200 serotypes can cause gastroenteritis related to Vibrio parahaemolyticus, serology is not an important test here. So whether Vibrio parahaemolyticus is present or not is enough. You don't really need serological testing after that. Okay. So it causes gastroenteritis, abdominal pain, vomiting, uh, but recovery is after a few days. So usually self-limiting. Okay. So Vibrio parahaemolyticus important because of its association with seafood. Okay. So next is the Vibrio vulnificus. It's an emerging pathogen. It also causes gastroenteritis, but it is important when you have people with compromised immune system, those with chronic liver disease, where the mortality rate can be up to 50%. So in America, the NIH actually recommends those with chronic liver disease not to take raw seafood because of this. Okay, because Vibrio vulnificus can enter the bloodstream and cause primary septicemia. Okay, so persons, and this is associated with raw oyster, shrimps, crabs, and shellfish, and this can cause up to 95% of all seafood-related deaths. So if you don't have chronic liver disease, uh, but if you have cuts in your skin, open wounds, you should also be careful not to go swimming, because when Vibrio vulnificus infects your wound, it will cause a rapidly spreading tissue destruction. So limb amputation or aggressive debridement is recommended, because they need to ensure that they remove the bacteria from your wound. So this is an emerging pathogen associated with raw seafood, and also for those who have cuts, open wounds, not recommended to go swimming. because this bacteria is common in seawater, even in Malaysia. For the isolation and identification of Vibrio, very straightforward. This is important. Alkaline peptone water. This is the enrichment broth that is used for Vibrio. So alkaline because most Vibrios live in the sea, so they do better at alkaline pH. Okay. So by using this enrichment broth, you are able to obtain most of the Vibrio in your sample. And then after enrichment, you can plate it on a TCBS. So TCBS is a good media, a standard media. Almost all Vibrio can grow on TCBS except two. Only about two species cannot grow on TCBS. The rest can grow. So TCBS is thiosulfate citrate bile salt sucrose agar. Okay, so sucrose is in the TCBS. That means for Vibrios that can utilize ferment sucrose, they will form yellow colonies. So that's how you can differentiate between Vibrio cholera and Vibrio para, because Vibrio parahaemolyticus does not ferment sucrose, but Vibrio cholera ferments sucrose. So on TCBS, Vibrio cholera will be yellow, Vibrio parahaemolyticus will be greenish. Okay. So of course, for Vibrio cholera, you do the O1 antiserum and then do the Ogawa Inaba antiserum. For Vibrio parahaemolyticus, you can also use alkaline peptone water. But if you use TSB, trypticase soy broth, you have to add salt. So for Vibrio parahaemolyticus, it is a marine bacteria. All your biochemical tests, you have to add salt, to make it up to 3% salt, in order that the results are reliable. Okay, if not, the bacteria might not grow well. So if you're doing IMVIC tests, make sure that you have 3% salt inside. Okay. So testing of Vibrio is also recommended for employees in the food industry. So those handling food have to be tested to ensure that they do not carry Vibrio cholera. Okay, so usually they do a rectal swab from the workers, and then you enrich it before you streak it onto TCBS. Okay, so enrichment is a procedure that we usually carry out whenever we are handling food and water samples, because the counts are expected to be low. However, when we are working from fresh stool, we don't really need enrichment because there will be lots of bacteria, lots of Vibrio in the stool sample, so we can immediately streak onto TCBS. Okay, any questions about Vibrio?
No questions.
No question.
Okay, so can we continue?
Yes.
Yes.
Okay, the next is Aeromonadaceae. The type species is Aeromonas hydrophila. Okay. Aeromonadaceae, family Aeromonadaceae. So it's a gram-negative bacilli, non-spore forming polar flagella. It lives also in aquatic environments, okay, fresh and salt water. It's an important species because it causes disease in aquatic animals and humans. So it is a problem in aquaculture. Okay. So this is an important bacteria, especially for the aquaculture industry. Aeromonas hydrophila can cause ulcers, tail rot, fin rot, hemorrhagic septicemia in fish, and red leg in frogs. So we also have an industry that farms frogs for the frog legs to be sold to EU countries. Okay. So in humans, consumption of food contaminated with a large number of Aeromonas causes gastroenteritis. Okay, so if you have an open wound and you enter water with Aeromonas, you'll get cellulitis or skin inflammation. So Aeromonas is important because of its threat to aquaculture. Okay, aquaculture and also aquarium fishes. Okay, any question? Any question about Aeromonas?
No, doctor.
Okay. Next is Pasteurella. Haemophilus is part of is under Pasteurelaceae. Okay. The most important is Haemophilus influenzae. You have other species. They are non-motile gram-negative rods, often encapsulated. It's important because the most important is Seroa B. So Haemophilus influenzae, Seroa B, HIP, causes most Haemophilus infections in humans. But you don't hear it now as often because there is already uh, a vaccine for this, and it is part of the immunization schedule in Malaysia. So if everyone actually undergoes the same immunization schedule, you don't actually will have a problem with this. Okay, but the problem is some people don't want to go for vaccinations. Okay, so it's a parasite of the respiratory tract, occurs in 30 to 50% of healthy humans. Because of it has capsules, so this is a virulence determinant. It protects the bacteria from phagocytosis and actually allows the bacteria to cause further disease. Okay, so it causes upper and lower respiratory tract infection, age 4 to 5. So it's a problem with in children. Okay, so what's interesting here is when you're isolating for Haemophilus influenzae, one of the identification step is actually to look for the satellite phenomenon. So on blood agar, you streak a Staph aureus. So you first you streak the Haemophilus influenzae like this. Streak a Staph aureus onto the blood agar. Because Haemophilus influenzae doesn't grow well on blood agar unless it has certain growth factors, the X and the V. Okay, so what happens is the Staph aureus growing here will lyse the blood. It will lyse the blood cells here and will release the V and the X factors into the uh, culture medium. So when there is a release of X and V factors in the culture medium, then the Haemophilus influenzae will grow better. So this is called the satellite phenomenon, where the Haemophilus influenzae grows better surrounding the Staph aureus culture. Okay, so this is a interesting fact from Haemophilus influenzae. So you also have Haemophilus ducreyi that causes tropical venereal disease, and also Haemophilus egyptius, conjunctivitis, infection of the eye. Pasteurella. So this genus is named after Pasteur, Louis Pasteur, because he found the bacteria Pasteurella multocida that causes uh, cholera in birds, in fowl. Okay. So Pasteurella multocida is a type species, causes zoonotic infections. So it has a reservoir in domestic animals and livestock. So it's a serious pathogen for cows, rabbits, and birds. It causes upper respiratory disease in farm animals. Okay. So they are able to grow on blood and chocolate agar, and it can be treated with penicillin or cephalosporin. So, so in 1880, so Pasteur found this fowl cholera. Okay. So what is interesting is he found that when he cultures the Pasteurella multocida after a long period in the lab, the disease loses its virulence. So when he inoculated it back into the fowl is not diseased, doesn't have the disease, but then it becomes immune. Okay, it becomes immune to subsequent infections by virulent bacteria. So this is the first, uh, not the first, but the proper experiment showing the importance of vaccination. Of course, the first person to truly made an impact in vaccination is, of course, Edward Jenner. Okay, Edward Jenner. You must remember that Edward Jenner uh, introduced small cowpox vaccination as a preventive protection from smallpox in 1796. So this is almost 70 years before Robert Koch established that microorganisms causes disease. So that's why this is a very important uh, discovery. So even before they know what causes smallpox, uh, they were already able to find uh, a way to vaccinate and prevent smallpox. Okay, so this is the so if you read up on Edward Jenner, you know that he's a very scientific person. Okay, so he heard that uh, people who had cowpox couldn't get smallpox. Okay, so he became curious and decided to test it out. He tested out on a total of 23 patients, 23 volunteers, including his own son. So he took cowpox material and then vaccinated the person's arm. Then the person will get cowpox, but will recover because cowpox is mild. And then after that, he actually uh, tested the volunteers with uh, smallpox, and none of them uh, were infected with smallpox. That's why he carried out his work on 23 uh, persons. He he noted the results properly. He explained properly, and because of that, when he published it, the physicians at that time in UK, in England, accepted it as one way to prevent smallpox. At that time, smallpox occurs regularly, annually, and about can cause death in about 20% of the population. So because of this, he actually saved a lot of people's life through this vaccination. So yeah, that's why Pasteur also stated the term vaccination in honor of his work, Edward Jenner's work. So nowadays, vaccines are still produced from avirulent microbial strains. So similar strains, but no longer virulent. Okay, so when you have this, when you stimulate the immunity in the person after injecting an avirulent strain, uh, the person will be immune to the virulent strain. So this is uh, the protection from disease provided by vaccination is called immunity. Of course, now we also use genetic engineering to only select the components of the virulent microorganisms. So we do not use the whole organism anymore, but we only use the antigenic components, maybe a protein, a surface protein, or something else. Okay, no longer the whole organism. But some, for example, the COVID-19 vaccine from China, they still use if I'm not mistaken, one of them still uses the whole uh, virus. Okay. So why we are divert why we're talking about this is because vaccination is important. Okay. So in Malaysia, we have a very good uh, vaccination schedule for children under 2 years old. Okay, proper vaccination schedule to prevent uh, against TB, against hepatitis B, okay, and against uh, the DTP, diphtheria, tetanus, pertussis. Now this is a newer schedule. So if you remember, diphtheria is Corynebacterium diphtheriae. Tetanus is Clostridium tetani. Pertussis is Bordetella pertussis. Polio is virus. Hepatitis virus. Haemophilus influenza type B. So these are all diseases that can cause mortality in children. Okay, serious mortality. So the mortality rate is high if you do not carry out the vaccination. So if you look at these graphs, these are from America because they kept their data well since the 1900s. So you'll see a continuous reduction in mortality rate from infectious diseases. So this is attributed to both antibiotics and vaccination. So without antibiotics and vaccination, the mortality rate will be very high. Okay, so antibiotics and vaccination has reduced the number of deaths tremendously. Okay, but the problem now is we are seeing an emergence of antibiotic resistant strains. That's why vaccination has to be continued. Vaccination is important because if the strains become uh, if the bacteria becomes more resistant towards antibiotic, we have to undergo vaccination in order to prevent uh, diseases caused by bacteria.